Welcome to Berry's In the Interim podcast, where we explore the cutting edge of innovative clinical trial design for the pharmaceutical and medical industries, and so much more. Let's dive in.
All right, welcome. We are back in the interim. This is Berry Consultants' podcast of all things statistical, all things scientific with clinical trials, medical decision making, and we have an interesting podcast today in that I don't know the topic. So my co-host on In the Interim, Kert Viele, is here, and he has a surprise topic to talk about today. Kert.
Hey, Scott. All right, so I want to start with a story. I went to a conference not that long ago, and standard statistical conference, people are talking about the data they had, all the methods that they've been developing to understand it, what they've learned from the data. And essentially all of these talks, they had a really interesting structure. So they get through all their methods and everything. And at the end of every single talk, it was ended with, if only I had gotten a chance to design the experiment, design the data, do this in advance, everything would have been better. I would have avoided all these problems and so on. And so I'm listening to all these topics. And when I get to my talk, of course, I started it after listening to all this, is I live in utopia, and then talked about experimental design. And so I get back in the car on the way home, and I'm sitting here thinking about, so am I really in the good place or am I in the bad place? Because one thing that's always impressed me about the last 20 years in experimental design is all of these methods that we're developing to understand data, causal inference, all of these aspects of how do we make inferences from data in front of us. We don't use those in experimental design. So we often have an idea that when we design a trial, we're to ignore every bit of data that's ever existed on Earth. If I go to my doctor and I ask, why are you giving me a drug? They're going to report lots and lots of studies that say this drug is good. If I go into an experiment and say, I want to use this data, I'm immediately told it's bad. And that depends on if I'm borrowing information. I got a drug that's targeted for a specific mutation. I have data on thyroid cancer. Can I use that for lung cancer? Generally, the answer is often no. If I want to borrow historical data from old clinical trials, Alzheimer's, we have hundreds of thousands of patients that have been treated on placebos. Can I use it? Well, there's problems with that, so on. Real world evidence. So I would actually define the standard debate that we've been having for the last 15 years is essentially, is data good or is it bad in experimental design? So does it lead us to a good place or a bad place? So I'd like to talk about where we are, how we got here, and where we think we're going. So I'm going to leave it up to you. I've surprised you with the topic and see what your reactions are.
Okay. Well, let's sort of figure out the topic and the interesting part of this. So when you say is data good or bad, do you mean that when I design an experiment, I'm saying I want to collect the following data and I can analyze just the data in that experiment, which we've been doing for a long time. Frequentist, rarely Bayesian, but we do that. You're saying by data, is that outside the experiment? Is there any room in that experiment for external?
Yes. So what do we do with essentially the totality of human knowledge prior to the experiment? Should that enter into the experiment in any way? And want to avoid this. I mean, there's lots of rhetoric we could do with this. If you want to say bad things about this, you talk about biases and confounding and everything else. If you want to say good things, you talk about totality of the evidence. But really, let's get at, what is going to lead us in good and bad directions? How do we decide when to do this and when to not?
Yeah, so let's lay out why not. Why? I mean, what are the reasons people give that we shouldn't use other data in our experiment?
Obvious answer would be it could lead us in the wrong direction. So the experiment might have been done with a different set of patients at a different time. There are key differences between that old data and what my current experiment is. If I use the new data or use the old data in my new experiment, I can get answers that are biased in certain ways. I can draw wrong conclusions. I can say drugs work that don't, simply because of the information I'm bringing in rather than the experiment itself.
So is this a frequentist issue? Is this that we calculate the operating characteristics of the new experiment? Type 1 error is only the new experiment. And if I use any data outside of my experiment, you can inflate type 1 error. You can get bias, all these bad terms in it. Is it a frequentist problem that data outside the experiment, all of a sudden, type 1 error means something very different. Bias means something very different. That I can't use stuff and be a frequentist?
So I don't know if I think there is a frequentist Bayesian divide. I think Bayesians are trained with the idea of collect some data, update your beliefs, collect more data, update your beliefs. And it's a natural thing to do to bring things in. But I don't know if I were to say that the problem is not being a frequentist in terms of do you care about long-term error rates, I would lay the problem at the feet of you have to have this 2.5% type 1 error in the worst case scenario. So in effect, we're playing minimax against nature. We have to assume that nature has deceived us for the last 25 years. And then in that case, we shouldn't use the data. So on the one hand, that delivers an immense amount of robustness. On the other hand, we're reinventing the wheel.
Yeah. So every time. So let's talk about a case where we would use external data, and we have. We've designed trials where we've used external data, and what that looks like, and what could be the potential problem. So suppose I've got results from another trial, a phase 2 trial about the relative efficacy of a treatment, and I bring it into my next experiment, and I use that as prior knowledge. And when I'm done with my new experiment, I combine them together. Or I bring in previous data on the control arm. I'm comparing to an active comparator, something that's approved, it's standard of care, and I want to run a new experiment of my new drug, and I want to compare to standard of care. There are lots of trials and lots of information about standard of care. Why do I have to run a 1-to-1 randomized trial, standard of care to my therapy, when I know so much about it? So I might bring in that information specifically into the trial. And both of those notions can cause issues in my new experiment, essentially if it's wrong. If that is different than my new experiment, statisticians are great at saying, well, boy, if that data was a little bit different, now your new experiment has type 1 error or bias or issues that if I bring in anything from outside the trial, it can cause issues. So that might be ways in which I bring in external data.
And there are certainly, there are cases where you are going to run into those problems. We've talked in examples in antibiotics, the development of resistance over time. We expect therapies to change. There are situations where we think that problem exists and we need to address it or not use the data. There are also cases where diseases are very stable over time, and those issues may not be as big a concern.
Yeah. So I'm very much on the side that we should be using external data. I think we go about this in a way that's just so strikingly conservative that it slows us down in it, that something may happen. I think it falls under the realm that science is hard. Incorporating this other information, be explicit about it, incorporate it. And the process of this is hard, but the notion that we don't use any of that information just seems strikingly wrong.
So it's an interesting question because it certainly can be wrong on occasion. So I wonder if as a society, and you asked about frequentist and whether the error rates are driving this, I think you can be a perfectly good and wonderful frequentist and use historical data. But what you're going to have to accept is most of the time, prior data is leading you in the right direction. If it's not, we should just cease science altogether because we have worse problems. But in any case, if it's generally leading us in the right direction, we're going to be running a few experiments that have, say, 3 or 4% type 1 error, and we're going to be running a lot more experiments that have, say, 1 or 2. And in the grand scheme of things, we're still putting a better mix of drugs on the shelf. More drugs that work, less drugs that don't. So I think we have to change our mindset to be frequentist in this kind of world.
Yeah, I'm struck by the idea of using that data. And in Bayesian, we do borrowing of that information. We do things that are dynamic. I'm struck by there are tons of trials that we use objective performance criteria. That it's a single arm trial and the drug has to jump a particular hurdle. That's a number. That number is always based on prior data. And we do that all the time, and presumably that's okay. Or you run a trial where your data comes from the new experiment, and that's your control. But the idea that we do something halfway between that we use it just strikes people the wrong way. Just do one or the other. And I'll give an example of this happening to me where, by the way, and if you listen to In the Interim a lot, you'll find that we very much respect the FDA. In the world, they do tremendous good and they're wonderful. It doesn't mean we don't run into hurdles. So we presented
so just to interrupt you there, I mean, we go to the FDA 100 times a year and have 3 major disagreements. That's much better than with our wives.
Yeah. So we provide an example that we wanted to go in with a new therapy. It was oncology. And the new trial, we didn't want to enroll 1-to-1 to a standard of care. It's easier to get patients in if they're more likely to get experimental treatment. We wanted to enroll 3-to-1, 3 to experimental, 1 to control. There's a good bit of information about the behavior of the control. So we were borrowing from previous trials. And let's assume that the prior data was a 15% overall objective response rate. Make it simple. But we're going to enroll 3-to-1. We're going to borrow on the control using that prior data that's centered on 15%. In the new experiment, because we're borrowing data that's 15% on the control, if the new rate in that new experiment is actually higher than that, we're pulling the control down, making it easier for the treatment to look better. And we present those operating characteristics. That was deemed to be unacceptable because you inflate type 1 error rate. The response from the agency was, use 15% as an objective control. And don't enroll any new controls, and you have to beat 15%. Of course, that we generally don't calculate the type 1 error if, in fact, the real rate in that experiment was 25 or 30% because we're jumping 15%. But it's almost the opposite Bayesian view that we know that answers 15% for what you're trying to beat. And if you can beat that, that's good. It's using a prior that's all focused on a single value rather than modeling it with some borrowing and recognizing the uncertainty, or enroll a whole control arm. So do one or the other, but it's entirely this experiment. But the idea of using borrowing or modeling seems to be harder to accept.
It's interesting. It's almost, you have to cross the Rubicon of what, are you willing to completely make this assumption or not make this assumption at all? And you can't test the assumption.
Right. And the other extreme of this, and I know this one gets at you a little bit.
Don't do it, Scott.
Yeah, that we talk about using real world evidence all the time. Digital twins or real world evidence. So for a control, we bring in external data, and people seem to be almost comfortable that your control is entirely external data. That's okay. But if you were to do something like use non-concurrent controls from your own experiment, that seems to be unacceptable, despite the fact that these are unbelievably phenomenal historical controls and better than any real world evidence you're going to get. Same protocol, you have the exact same data, the same quality. The only difference with them is time. People seem really uncomfortable with that, but yet at your conference you went to, there were probably 10 talks about using real world evidence.
There are different legal reasons that real world evidence is being handled in certain ways right now compared to other, but FDA is not it's not a homogeneous organization. There are lots of people. The academic community is many people. Industry is many people. But I think if we're talking about where we're going, one of the key aspects of this is a hierarchy of what's more or less dangerous. And I'd actually like to see us spending more time on Alzheimer's trials. We've got dozens, hundreds of these. So how much do the control arms differ from trial to trial to trial to trial? If we have hundreds of pieces of trials, we should be able to estimate this. We're statisticians. We can estimate a variance parameter. Let's get an idea about what it is, and that variance parameter translates into how much we should borrow.
Yeah. And you mentioned a key point to that. If you're enrolling some new controls, you actually get a comparison. You get the controls in your experiment against the previous controls, and you can judge some level of similarity between those.
Can you talk about that a little?
Okay.
Why don't you talk about that a little bit more about just the details? Because I think there's often a sense of, the fact that there is a hierarchy between a single arm trial, dynamic borrowing, static borrowing. Get an idea about what those methods are and how they mitigate those risks or amplify them as the case may be.
So the extremes on this, that I can run an experiment where I only include data in my new experiment, 1-to-1 randomized control to treatment, and I'm only going to compare those two arms in that. I could do the exact, very much extreme from that, where I enroll only my treatment arm. And I want to know, is the treatment arm behaving, better than, is the treatment benefiting these patients? I would have to figure out a way to create a control arm. I could use entirely historical data. In that far extreme, I have no ability to know whether that control rates I'm using is reasonable. If suppose I do something halfway, that I enroll 2-to-1 or 3-to-1, I use the controls in that experiment, but I also use external controls to reinforce the control. Now, I'm getting new controls so I can judge their similarity to the external controls and statistically model that similarity using the external data to strength if they're similar and not if they're not. But I do get evidence about their similarity, and it's different than this extreme case where I only use controls and I have no idea whether they're good or not, as a middling ground to this.
And I think, that's really what's going on in the academic literature right now in terms of trying to figure out the best ways to make that assessment, minimize the risks, amplify the benefits. We're talking now more about covariate matching between studies. So not just looking at all the historical data, but which patients best match our current enrollment. None of that, of course, is going to be perfect. But I think, it's the question of, do we generally add information, or are we taking a risk? And we should be looking as a society, if we do these kind of trials over and over and over again, do we get a better set of drugs on the shelf?
Yeah, and part of this is statisticians are really smart, and they can figure out how something can go wrong. That the historical data is not the same as the new controls. We've got problems with the analysis there. We've got potential type 1 error inflation. We've got potential bias in the estimate of the treatment effect. And statisticians can figure this out and say that, this is bad. But if we have to do experiments where we have to enroll 1-to-1, that experiment is bigger. It's more patients on control. We take less shots on goal. We learn slower. It's the whole industry is slowed if every time we have a question, it has to be only in that experiment as opposed to using what we know scientifically to reinforce that, making a more efficient trial. We can look at more treatments. We can treat fewer patients. We can do this cheaper. So there is a huge societal, I'll call it societal, a whole medical field issue. If every time we have a question, the only thing we can do is look at the new experiment, it seems like there's a huge waste.
Well, it's like data. We design an experiment, data comes into existence. There is this bright, shining moment of about 5 minutes where we can do something with it, and then we're meant to put it aside, at least with regard to future experiments. Obviously, people are going to reference it in deciding treatments and so forth in the meeting. But it's interesting. It worries me as a statistician. I feel deep-seated guilt actually putting data aside.
Yeah. And again, it's hard, but there's huge ramifications to not doing it in that way. And I think there are ways of doing it very well. You can be prospective in your new experiment. The analysis plan is completely written down. It's explicit. When you present the results, you show the results just in the experiment. You show what happens using the modeling of external data. I think we can do this really well. I think, by the way, this is changing a little bit. We're seeing this even a somewhat similar issue where you run basket trials. There's a lot of reticence to, we have 4 kinds of patients, and we run an experiment of control to treatment, of using the data in one of the subsets of patients to help estimate another subset of patients. That gets people really bothered. And that's been done more and more, I think. It's a very similar type of issue, and it's sort of striking, and we're in trouble if we don't do that, but it has similar statistical ramifications.
Well, and it has tremendous implications towards design, because if you can't borrow, suddenly sponsors have an incredible incentive to claim that their patient population is homogeneous so that they can pool everything together and maximize their power. If suddenly you say, as soon as you say there's 2 different groups of people, you have to run twice as many patients, no one's going to want to explore those kind of questions.
Yeah, so it's the same bizarre thing that happens, that we run trials that are single-arm trials where we use historical control 100%, or we don't use it at all. And you're describing exactly what happens in trials. We run a trial and you do a common estimate across the entire population, a single estimate, which is full pooling. It's the exact same mathematically of pooling populations all together to get a single answer. Or we run separate trials and we call them population A and population B, but we only estimate those individually in A and B. Now we want to do something that's a middling thing between that. And by the way, we actually get to observe in the experiment similarity of effect, that we shrink estimates statistically, that isn't full pooling and it isn't separate, and that people struggle with that. It's either you have to do all pooling or completely separate populations. And it's just, it strikes me as just bizarre.
So let me ask you about one other thing, which I think is a real oddball in this conversation. There is a situation where we routinely combine data across different studies, and it's often viewed as the pinnacle of evidence for clinical trials, which is a meta-analysis.
I have no idea. Meta-analysis. Okay. Yep.
So where do you think meta-analysis fits in terms of the assumptions and how it fits into this debate?
Yeah, I think meta-analyses are incredibly valuable. The modeling makes a ton of sense and incredibly important. I think people overweight single trials and the totality of evidence. Now, not every meta-analysis is every meta-analysis. And I think people sort of think meta-analyses are bad because there are some that are clearly biased. There's publication bias. There's availability of data bias. So not every meta-analysis is bringing quality data that's free of these issues. But there are many circumstances where we have data on essentially every patient that's been experimented on. And combining this together, I think, is hugely valuable. By the way, I think the idea that we run two separate independent phase 3 trials and we analyze them separately is stupid. I mean, it's stupid that we would analyze them completely separately and we need both to be 0.05. Combining them together into a single inference is better.
Well, this is the standard.
In every way.
Is two 0.049s as good as a 0.051 and a 0.0001 on the p-values? Clearly, you'd want the latter.
Yeah. And so I think of a meta-analysis the same way. I would do a meta-analysis of my 2 phase 3 trials for what does it tell me to the answer. So, those techniques, they tend to be Bayesian, but largely the availability of data. And there are some where we know there's missing data, we know there's issues with it, and we would not take those at a high scientific degree to that. And that's where the science parts comes into it. And by the way, these are abused, where people present data, it's biased data, they're ignoring huge amounts and they call it a meta-analysis, and we think that's bad science. So we've got to, at some level, be able to judge good science, quality data, meta-analyses, which I think are more valuable than individual trials there. And then there's bad science put together that we recognize we wouldn't use that information.
And we would certainly say that on any kind of borrowing. Bringing together data that doesn't belong is bad. You can borrow in a way that does generate horrible biases and will generate bad conclusions. But we also are talking about what can be done well with experience. We're running out of time. Why don't you close us out?
I think this, by the way, is the future. I think as we learn more and more about diseases, we get more and more subsets of them. We're getting more and more quality data available from other clinical trials, the sharing of data from clinical trials, the sharing of other types of resources. We're going to have an explosion with medical data. The idea that we don't use that in our new experiments, if we don't use that in valuable ways, we're running worse experiments. So a wonderful topic, and I think it's certainly the future. And so here we are in the interim. So till next time, appreciate it. Thank you, Kurt.